Bacteria quorum sensing by Anyi, Ting, Karl, and Raj. Bacteria, what exactly are they? Well, bacteria are microscopic unicellular organisms that have been around for billions of years. They carry a few genes and genetic information within the single strand of DNA that they have. Bacteria thrive by taking in nutrients from the environment and reproduce via binary fission, where cell elongation and division take place. Although we think of ourselves as humans, we are more bacterium than human. Our bodies have 10 times the amount of bacteria cells than we have human cells. As for genetic information, humans have 30,000 human genes and 100 times more bacterial genes. So theoretically, we are only 10% human and 90% bacterial. We have a symbiotic relationship with these bacteria, which are vital to our survival. They protect us from harsh environmental conditions. They allow us to assimilate food and enhance our immunity against harmful bacteria. However, there are also pathogenic bacteria that pose a major threat to our well-being. Hence, it is crucial to understand how different types of bacteria function. Moving on, we're now talking about the symbiosis between Hawaiian bobtail squid and Vibrio fischeri. They share a unique relationship. During the day, the squid sleeps buried in sand. Once night falls, the squid ventures into the ocean to hunt. To hide itself from predator and prey alike, the squid uses its own invisibility cloak, a colony of Vibrio fischeri that emit bioluminescence. The squid uses this light to mimic moonlight, effectively concealing its silhouette and escaping the eyes of other organisms. Researchers found out that bioluminescence is only emitted from a concentrated cell suspension, while in a dilute suspension, light is not emitted. This is because Vibrio fischeri and other bacteria have multiple enzymes within their bodies that produce signaling molecules. These molecules are released out of the cell and bind to the receptor protein on other cells of different bacteria. When a sufficiently large number of this molecule is present and detected by other bacteria, it indicates that a critical cell density is reached and they can carry out the collective process they intend to, for example bioluminescence or bacterial infection. This method of chemical communication is called quorum sensing. Moving on, there are two main types of bacterial communication: intraspecies communication and interspecies communication, both of which can be used by all bacteria. Intraspecies communication. Bacteria cells only talk to the same type of bacteria. These cells will secrete a specific hormone that only binds to the complementary receptor protein. Interspecies communication facilitates communication between different types of bacteria. It involves a second enzyme that secretes a second hormone, which has its own specific receptor that is common in all bacteria. One of the important things bacteria can do with quorum sensing is to initiate pathogenicity. The problem of increasing antibiotics resistance is becoming more prominent and the world is running out of effective antibiotics. So, there's a need to improvise and make new behavioral modifications to those bacteria by disrupting the process of quorum sensing. A way of making new kinds of antibiotics is by making false signals to make bacteria unable to talk or hear, so that they cannot communicate with or sense other bacteria. By making such improvements, the bacteria will not know when to launch virulence and therefore will not cause disease. There are two ways we can do this. One, by targeting intraspecies communication. The purpose of this method is to make species-specific or disease-specific anti-quorum sensing molecules. Another way is to target interspecies communication. These can be used as a broad-spectrum antibiotics that work against all bacteria in the future generation. We can also enhance quorum sensing to, in this case, help the good bacteria in our body. We will be able to use it to help bacteria by enhancing how they talk and hear each other, and make bacteria do things that we want them to do, to help us better than they already do themselves.